introduce Alejandra Medina-Rivera from the National University of Mexico. And Alejandra.
Thank you. So today I will talk about more like hard work and little results because we're still building these resources. So yeah, this goes a bit more into the stubbornly small data sets. So we are not really in the big sets. So, you know, patient registries are very valuable. A lot of you use them through the UK Biobank, the All of Us initiative here in the States. And there's also like big resources like the Twin Registry in Australia. And you know how useful they are. These are organized systems that have all the data stored and that have recontact, so you can see what happened to these people after years, so you can build longitudinal studies, and that have genomic information that is accessible. So, but there is an issue with these resources and this diversity. So most of the genomic resources that we use, they basically are built using European ancestry descent samples, so people that live in the global north. And there's some representation of Asian populations and the African population that also have quite an effort. But for example, Latin America, or what's sometimes labeled as Hispanic populations, well, we are poorly represented with like less than 2% of the samples in general, not only in these resources, but in general, in genomic data sets, in genetic studies. So these are poorly represented. And furthermore, these samples are all categorized as equal, while there is a different socio-demographic history along the continent. So this is an issue. So what are the efforts in Mexico? So how, and this is a question I have a lot, how are electronic records shaped in our country? So 70% of the people in Mexico are affiliated to a public or a private health system, but we have 5 different systems. So 50% of the population would be, of this 73% will be associated to the Mexican social insurance system. But there's also something called the wealth-being system that is more like people that don't have officially declared jobs, so people that are like poor or that don't really have a social security system through their jobs. And however, 33 million of people in the country, we are a 120 million people country, have no services. But in general, like 50% of the population will have no way to get any health service, and not even within the private sector because they are really far away or they cannot pay for it. There is an official norm that was issued in 2012 that rules how electronic records should be settled. And the issue in Mexico is that as we have 5 different systems and there can also be subset within regions, there is no way to have one sole electronic system, and there will be hospitals that at the end will drop them. That happens a lot at the end. The electronic system is not efficient. There is no internet connection. Sometimes politicians take decisions that are not feasible. So you cannot set up an electronic record in a place where you don't have electricity. So at the end, they will go back to paperwork. So this makes any usage of this data barely impossible. So what is the perspective in Mexico? So currently, actually in 2022, there was the release of these 2 major genomic resources. So there is the Mexico City Prospective study that is based on samples that have been taken from the 90s. The system of recruitment is based on 2 regions within Mexico City. So one is called Coyoacán and the other one is Tapalapa, and they have an important difference in socioeconomic status within the 2 regions. So one is middle upper class and the other one is low middle class. And so what they did is that they went to their homes, and this is a geographical based sampling. So they can only go again to the same houses, but they cannot trace back the people. So if people moved, they will lose their recontact. So they did a medical survey with some anthropometric measurements like weight, height, BMI, and like glucose in blood. And they did exome sequencing, and this is 150,000 people. As I told you, recontact is based on address. So in 2015, 2019, they recontact people based on this. So they could only recontact 10,000 participants because people have moved. Particularly Coyoacán has been heavily gentrified in the last 10 years. So people are moving out because they can no longer pay living there. Iztapalapa a bit less, but this gentrification is an issue across the whole city. The major publication was recently released, and this is an effort led mainly by Oxford with the collaboration with the National University in Mexico. So all this data is in Oxford in the UK. There's another research that just also came out, the MEX Biobank survey, and this one is led by a group in Mexico, in Langevio, by Andres Moreno. And these are samples from the National Health Survey in the 2000s, so these are blood samples. They have now genotyped 6,000 people. In total, there are 40,000 samples, and they have a general health survey. Unfortunately, this was done with ethics without recontact, so they cannot trace back these people and know what happened to them. There are no autoimmune diseases questions within this survey, and it's also an issue with this one. And there's also no information of mental health status within this cohort. But nonetheless, these two resources are key now in Mexico to at least know the allele frequencies in the population, because that's something we didn't even have. So challenges, recontact is an issue in our major studies. So there's no way to have like further stages in the research or do longitudinal research. There is an issue with centralization of the data. So one of our major resources was built only in Mexico City and only into regions in Mexico City. So that hinders the nationwide capabilities of this research. There is a continuous failure with the collaboration engagement with the medical community. So medical communities in Mexico are a bit more worried about saving lives than taking data from patients or samples for biobanking. So there's also an issue of lack of trust towards the scientific community, and this has been documented through the National Institute of Statistics with the survey on perception on science and technology, where people have reported not trusting in scientists because they know too much, they have access to a lot of money, and they don't report back their results, which is not a lie. In Mexico, there is still a building, So in Mexico, people are not used to report back to participants to at least tell them what happened with the data they gave or what results they got. So there is a lack of trust also because of that. So those are the challenges we had. And so we decided to start setting up 3 different registries, the twin registry, a registry for Parkinson and a registry for lupus. And the idea is to have comprehensive medical history surveys. We have ethics and consent that contemplate being recontact for follow-up studies. We have ethics approved for biobanking. So in the 3 projects, we are building a DNA data bank. We have mental health, cognitive assessments, and lifestyle surveys included. And we are really pushing this to be a nationwide recruitment perspective. So we're really trying to get people from other regions in the country and not only Mexico City. And we are working a lot in public engagement through continuous communication with participants using social media and Facebook Lives and Instagram and TikToks. So I will talk to you about these 3 surveys. So within the Mexican Twin Registry, we launched this in 2019, just before the pandemic. So we were able to sample some participants in the Twin Festival that this community has every year. And we are collaborating with the National Multiple Birth Association. We do data collection through RedCap. So most of the data collection is all purely online. People can get into our Facebook pages and Twitter and just register through that. We keep recontacting them so they can fill in other surveys. So it's always a work in progress. So at the beginning, we had only on-site sampling, but well, now with COVID, we really launched social media campaigns and also traditional media, because there's a lot of people that do not really have access to social media. So, and we do a lot of public engagement in the Twin Festivals, and again, with social media campaigns like this, like where we struggle our missions, or basically how to get into the registry and log in and do the surveys. So initially, the surveys have been selected to match the Australian and the United Kingdom twin registries, because we also want to enable comparisons of data, because otherwise like we will be alone. And we have a strong focus on mental health and cognitive development. So we do a lot of surveys on that too. And we got money from the National Science Council to do functional magnetic resonance of the twins. And this is up to now, we have 100 pairs that have been also biobanked. So we have DNA samples for them. So now we currently have more than 3,000 people registered. However, not all of them are sets. So we sometimes have only one twin of the set. So still that hampers a bit the analysis, but now we have almost 600 pairs that enable us to do some analysis on heritability of some traits. So most of the age of the registries is between 20 to 30 years, which makes sense because we are heavily based on social media campaigns. So these are the people that access social media. And mostly we have females as well. And this is a trend you can see in a lot of biobanks where women are more likely to participate in research. So that's a bias we have. And also we now have more or less the same number of monozygotic and dizygotic twins. It's like about 1,300, 1,200. So yeah, now we are able to do some analysis with this data. And this is just like a bite of our progress here. This is done by the postdoc Talia Roman, who has been analyzing the data of reported myopia and astigmatism in this population, and using an ACE bivariate Cholesky modeling, she has found that there is this is the percentage of her heritability of myopia and astigmatism in the Mexican population. So now we're able to do some analysis in our data set, which just started. This is like from this year, we reached a number of participants that allow us to build these tests. So this is the team, like these projects require a lot of people devoted to it. So this is a strong collaboration with Sarel Alcauter and Alejandra Ruiz, also from UNAM, and Miguel Renteria at Queensland University. And these are all the students that are devoted to this project, with the collaboration with the National Multiple Births Association. So the other project we have is the MEXPD, the Mexican Network for Parkinson Research. And this is a project that is part of the large PD. So this is a Latin American survey of Parkinson that is trying to collect data to build the biggest GWAS for Latin American populations for Parkinson. And this is also part of GP2, the Global Parkinson Genetics Program. So in this case, the enrollment of participants is only through neurologists. So this is important because in this case, we really wanted the phenotype to be well characterized, well done, well registered. But there is an issue because we have a shortage of neurologists with a valid certification. So this hampers our capabilities to sample people in some regions where we don't even have a neurologist that can do the assessment. The surveys are designed to match also like large PD, but also the American Research Consortium of the Genetics Parkinson Disease. We have funding from the Michael J. Fox Foundation to genotype 2,500 patients and 2,500 controls. And we have financing from the American Parkinson Association as well to do deep phenotyping with cognitive decay assessments and mental health surveys for 500 patients and 500 controls. And up to now, we have 13 clinical sites across 8 different regions on the country. Sorry, it's not so easy to see here. And we have also 2 sites for control registries, which has been really challenging because we found out people in Mexico are not really likely to participate as controls. They need to have an incentive, and the incentive is usually being sick. So we are trying to change that through a lot of campaigns, trying to help people understand why this is important. We have 3 sites in Mexico City, so we again see this bias, but we have been able to recruit people from other regions like Jalisco, that is also a big state in Mexico, and Querétaro, that is where our institution is based on. So we also do, so we have 13 neurologists that are actively registering patients. We also have sampling days calendarized now in certain sites because, so particularly when the neurologists come from a public institution, they don't really have the time to do the assessments or the surveys or to even do the swapping. Everything, it's really tight there. So we have like Sundays and Saturdays where the patients are calendarized so they can come and we can do the assessments ourselves, but the neurologist helps us doing some of the evaluations. We have public engagement sessions through Facebook, and we also do like public appearances through the universities. And we also have an open campaign for control recruitment, where we try to go to health fairs or like festivals in the States so we can get some controls in those regions. So up to now, we have, well, this is a bit old now. We have almost 200 cases and almost 200 controls. So this is from May. And my lab is mostly devoted to understanding the effects of anxiety and depression within Parkinson in Mexico. So this is led by Paula Reyes, who's a PhD student. And we have also now data for different surveys that are focused on anxiety and depression, and also other general mental health issues like the CCL90R. So we have, again, in particularly in cases, we have more males because also Parkinson is more present in males, but you can see there is a bias on the controls because most of the time is the wife that participates at control. That's the easiest recruitment. But we can already see a trait on anxiety and depression within the Parkinson population where they will have a higher score in these surveys. So this is also a tease of like what we're doing now in the lab. And honestly, these results are from last month that we hit the 100 patients, then we could do some statistics. Again, this is a very large effort. So this is also with Sara Alejandra and with Miguel. But we work also with large PD, with Ignacio Mata in Cleveland Clinic. And we have all the neurologists that are really helping us pushing this out. And also the NGOs that are communities of patients. So that's also helping us getting a bit more of traction in the recruitment. And we have a social media team as well here. And regarding the Lupus project, so I don't know, I probably shouldn't know, but there is a high prevalence in lupus in diverse ancestries. So actually Africa is the one with the highest prevalence, but there's also a trend in Latin American population. And also there is an issue because, for example, in Mexico, the official reported prevalence is 20 people in 100,000. However, there were some prevalence studies within some regions by some of the national hospitals. And there was one that found that it was 70 over 100,000 inhabitants and also 90 in Oaxaca. So we think that this number, it's a bit biased and probably is not well calculated because there is not a national effort or an epidemiological surveillance on this disease. Yeah, sorry, and it mostly affects women. So we launched this registry in May 2021, so it has been up for a year. The initial recruitment was only through social media, and it was based on self-report status and self-assessment of clinical manifestations. And this was because of COVID. So it was really hard to get people into the hospital. Patients completely stopped going to the doctor. That actually you can see now in the outcomes and in the clinical manifestation they have because they are doing worse because they haven't seen a doctor for 2 years. Now that they are coming back, we see this trend, and now we can document it, which helps a lot. So we started with social media campaigns and with a close collaboration with patient communities. So we really work together with these patients. They also suggest surveys because they want to learn more about their diseases. And we now have 5 rheumatologists that are recruiting actively patients, and they really joined in the last year when they saw the relevance of this project and that they could also use this data for their own clinical research. So currently we have more than 1,500 patients. Here I show you the analysis of data for 700 patients that completed the socioeconomic status survey and the medical survey. So we mostly have women, which reflects the disease prevalence and the presence in women, half of them have a scholar degree, a bachelor at least, and most of them have been diagnosed within, they are about 36 years old, and they are mostly between 29 and 44 years old. And half of them are actively working. So this is a disease that also hampers the quality of life during the productive years of the person, where they should be working, when they should be finished school. So most of them were diagnosed around 27 years old. They take anti-malarial drugs and glucocorticoids. And this is what I was telling you about the different systems. So most of them are treated by the National Social Security system, but 30% of them are treated in the private system. And this is another state-based social security that you get when you work in a federal institution. So most of them, they are treated in the public system, but we have an important part of them being treated in the private system. 6% of them, of the registries, were pregnant at the time they filled in the survey, and 40% of them have close relatives that also have lupus. So this is this was notable because, like, we know there is heritability trend, but we didn't know that it could be this high. And 24% of them have damage to the kidneys. So this is currently the regions that we are covering. So we are heavily covering Querétaro, that is where my institution is based. And there is this big community of lupus patients that is called Lupus Omics that is based also in Querétaro. But there is also another one that is Lupus Morelos, that's also heavily represented. And this is also this effect in Puebla that is just, those are two states that are together. And you can see that we don't cover all the regions, or that are really few patients in those regions, but we also have a map of the registered rheumatologists in the country, and it totally matches this map. So if you don't see high number of patients, it's just because they haven't been diagnosed. There is no doctor to diagnose them. So we already did some analysis on the socioeconomic status of our patients. So most of them are what can be considered middle class, and they spend most of their money in food, communication, transport, education, and health. And we already have some analysis of the comorbidities in our patients. So we mostly see no other diseases, but we do see some hypertension and thyroid diseases. And these are the principal clinical manifestations that we see in our patients. So we see heavily arthritis and alopecia. And actually alopecia is something that is very worrisome for these people because, well, it's how you look, and that matters for them. It's mostly women that are losing their hair. So they are really worried about this. So we did an analysis of the quality of life, and this is using as control group the twin registry. So we can see that there is an effect on the lupus patient on their perception of quality of life, and in particularly in the health domain. So they really perceive that their health is lower, but it also affects how they perceive their environment. So they think their environment is poor. They also perceive low physical health, low mental health, and that this affects also their social relationships. So this is soon to be published as well. And within this project, so I'm more a genomic scientist, and I like doing regulatory genomics, and that's more my lane of work. So we got some money from the National Council of Science, and now we are going to do a project of allele-specific expression patterns in systematic lupus during tolerogenic induction of dendritic cells. So we are getting 30 patients and 10 controls now, and we are doing this differentiation from monocytes to tolerized dendritic cells and challenging them to see whether they are able to stop an autoimmune response. And we are doing this because tolerogenic dendritic cells are a proposal of immunotherapy for autoimmune diseases because they could help turn down the autoimmune response. So currently we have recruited 15 patients and 6 controls, and we're actually able to differentiate the cells. And this is work done by Ana Laura, who's a PhD student in the lab. And we can already see with these 15 patients that we have changes in the presence of some of the protein in the membrane like CD11 or the HLA, and there is a difference between the patients and the controls on how tolerized dendritic cells are being developed. So I want just to thank all the people involved. Again, here also Alejandra participates a lot because the mental health part is very important for this. We also have a team of rheumatologists that have joined and are helping us recruit patients, but also to design new surveys. And well, CONACYT that is paying for part of this. And now, thanks to everything we learned, we are launching also the Pediatric Lupus Registry. And this is going to be led by someone else because I don't have any more time. So this is Claudia Gonzaga and Talia Diaz-Prieto. She's a rheumatologist at Hospital Texsalud in Monterrey. And Claudia is now an assistant professor in my institution. And she's launching the Rare Diseases Registry. And furthermore, thanks to everything we learned, now we are also part of the Chance to Convert initiative, and we're going to do single cell sequencing of immune cell types across different countries. So we're going to recruit 120 people from each of the regions. We are going to build an atlas of Latin American immune cell diversity, and we're going to work on building regulatory networks and the effect of variation in transcription factor binding. And well, we're biobanking a lot of the samples to be able then to do TCR sequencing. And this is a team that has been assembled for this. So in summary, we launched patient registries in Mexico, and it is a challenge, but it's also very rewarding. People are really thankful this is happening. Patients contact us, and well, it's something that you feel that is something useful you are doing for your country. So people engagement is key for this, and these researchers can have great impact in the research in the community because they are not only for us or for me or for my group. This is something that has to be available for everyone. And this infrastructure can be used for new registries like the one I just showed you. Yeah, this will also help increase diversity in genomic samples, but we still have challenges. So for example, the large PD data will be centralized using a system called Terra that is based in Google Cloud. And this sounds super cool if you have a way to pay for this. So I'm able to download my data. So the Mexican data I can download and have it in my servers, but I cannot access the data from the other regions of Latin America unless I have access to Terra. And this is something I cannot pay for because my university doesn't allow using credit cards. So that's an issue. And this is something that is not even talked about. So this is again going to be very advantageous for people in the global north that have systems to do this. But for Latin America, and this is not only my case, but the other teams see the same issue. It's hard. We have a lot of issues with corruption. So the regulation on how you can spend the money is really tight. So you don't steal anything, but you can also cannot also pay anything. So this is going to be a challenge that we have to find a way to get through. So thank you for your attention.
Thank you, Alejandra. I think we don't have time for questions, so maybe all discussions should be moved to a coffee break. And let's welcome our next speaker, Aaron Halperin from Option Labs and UCLA.